Yes, I think. I think I can hear myself. So I seem to be online right now. Hello, everyone again. Hi, Oliver here. I always introduce myself with hi, hello, Microbehunter here. And welcome again to another Saturday microscopy live stream. Yeah, I see that a lot of people have already joined in, and more people keep joining in. Yeah, and what I usually do at the beginning, I wait a few minutes for more people to join in, and of course, I wish everyone around the world, yeah, a nice weekend. And have to tell you, some of you I've seen really are staying up quite late, quite early in the morning, even just to be able to watch this video, this live stream. I have to tell you, I feel very honored. Thank you for that. But of course, the video will remain online later on as well. So if you're basically feel like that you're too tired, maybe, then of course you can always watch the video later again. Yeah. So. Just a little bit of housekeeping information again at the beginning. If you're new to this live stream, the format is as follows. During the first hour, I usually show you a few things, and you have the possibility to write comments in the comments section on the side. They can be on the topic basically that I'm talking about, but of course also some other microscopy related questions are of course perfectly fine and interesting. We're going to do a little change maybe compared to the previous videos. Several weeks ago, I used to always answer the question as they appeared in the live chat. And then there was a suggestion that basically I should answer the question at the very end of the live stream, basically that I just not interact with you. But then questions started to appear on the things that I actually mentioned, and then it's kind of difficult to respond to those questions afterwards. So I think we're going to try a new system today. I'm going to try to answer those questions that are related to the things that I show you. I'm going to try to answer them right away. And if you have some off-topic questions, then I'm going to answer them at the end of the live stream. So we're going to split this up. And however, I do have a request. Please do indicate that the question is actually directed to me, because many of you are actually also asking questions and chatting among each other. It's perfectly fine, but it's a little bit difficult for me then to actually also be able to find the questions. I have to scroll around, and then I might overlook something here. So I would like to request, maybe you can indicate this and you just say at Oliver or at Microbehunter, so that I know that you're talking to me. And then maybe you can also say that if it's an on-topic or off-topic question. Yeah, so maybe you can say on-topic, then I know to give those questions a little bit of a priority. Okay, I think let me quickly check again before I get started. A lot of people started to join in already. That's nice. I've got 45 people watching right now. Usually takes a few minutes at the beginning. What I'm going to do now is have a very quick look through the comments already. So yeah, hello all around the world. I used to send greetings to every individual country before, but I'll do this now collectively. Yeah. I'll get started. Okay, and I see that there are already a couple of questions here. That's fine. Yeah. Hello from all around the world. So what you can see here in the background over here, right? Yeah, and everywhere. That is basically a live view from a microscope above, and these are diatom frustules. Frustules. Yeah, that's how you actually call it. They're basically the silica shells of diatoms. Diatoms are related to the algae, and they have a shell made of silica. And today I want to show you how you can concentrate them and purify them. And I was, yeah, I would say not 100% happy with the result, but I would say that maybe I'm a little bit like 90% happy with the results. So it's still a little bit a work in progress. I still want to show it to you because one of the reasons why we're doing the live stream is because those of you who have a microscope and who are interested, you might want to try it also at home. And maybe you have a variation of the method, and then you can also share that, of course, in the comment section below. So I would like to quickly tell you a little bit more of the background of why I'm actually, why purify them and what's the whole point. When you have a sample from a water with algae, then in many cases you find diatoms in between there, and I've actually prepared a video to show you. Okay, because I'm going to also show you some stuff live from the microscope, of course, but just for the ease of demonstration, just prepared a video here. And what you see over here, and that's the nice thing, is I can also pause it, and I have an arrow as well. Okay, and what you're able to see here is of course these clusters that you see here. These are algae, I don't know, bacteria, sticking together. And then sometimes you see those, these here. These are diatoms, obviously. You see in the center here, these are chloroplasts, and they do photosynthesis. And this seems to be like a sand grain or something. And in many cases, those diatoms are sticking together, clustered around, like over here, and difficult to see. And I guess they have some kind of maybe a polysaccharide layer around them that kind of makes it sticky. But you see, they're not very clear and visible to see. And you can also see here in the background, you don't see a lot of them. So they're also not very concentrated. So what I want to show you today is I want to show you a method how you can treat the sample so that you get those diatom shells at a much higher concentration. Right, and this is what I have done. It requires quite a bit of patience, I have to tell you, and it didn't work all the time with all samples. I would also like to show it to you. That's why I say I'm only about 90% satisfied with the result. Yeah, so that's basically the thing that I would like to do here today. Yeah, here you can also see that there are many of these diatoms are kind of hidden here in clustered around in here, sticking together. Not so pretty, right? But I would like to now remove everything here and just to have the diatom shells. Yeah, so. So this is I'm skimming through the questions, and I'm just going to answer those questions now that are kind of a little bit on topic, okay? But please feel free to also ask other questions. I'll respond to them later. Yeah. So I've only very rarely found diatoms in water samples from ponds, rain barrels, and puddles in the forest. Where can you find more of them? Yes, I will actually show you where I found plenty of them. You will not find so many of them in the free water sample. What you have to get is, and I'm going to switch over now back to the desk view. You got to collect something that I call pond scum. That's the stuff floating on the surface of the water. So you actually need to collect some solid material. You can also find quite a few of them sometimes in the sediment. So if there's some fine sand or some stuff, organic material on the bottom of the pond, then you can also find them there. And many of them actually also grow on the surfaces of rocks. So you have to scratch them off. Yeah? And this is basically what I've done here. I mean, I can actually open the tube here. There's some kind of green scummy stuff floating in here, and most of the things here, however, are not diatoms. Okay, so this is basically contains so many other things, algae and so on. And what I want to do now is I kind of want to remove all of this other stuff so that I only have the diatom shells left over. And the procedure requires quite a bit of patience, but it's not difficult. And that's what I just want to show you today. Okay, so what I just want to tell you is you will not find them so much in the free water because they actually sink to the ground, right? And so you have to kind of collect some solid material from somewhere, and many of the diatoms actually will stick to a surface. They will grow on. So if you have some rocks, for example, that is kind of slimy in the water, right? You touch them and kind of have this slimy layer. And these are basically very often cyanobacteria and also diatoms growing in this biofilm. Okay, so that's actually what I recommend. Okay, I'm just skimming always the questions when I make a pause. So, and what I would like how do diatoms divide? Actually, this is actually quite well studied. They will divide, and what happens is I don't know if I can show this to you somehow. It's actually have like a box, so to say, and then the other half of the shell kind of grows in it. It's kind of nested in it. Yeah. So So this is somehow how they divide it. I read this somewhere, but it's kind of a little bit for me difficult to say. So with every division, the diatom shell becomes a little bit smaller, and then it starts to later on regenerate somehow. Yeah. Okay, so this is basically something that I would like to mention here, and I'm going to explain you the theory first before I actually show you what I've been doing. So if you have a sample, then it contains a lot of organic material like algae, like for example the green stuff here that I actually shown you. Also the inside of the diatoms. Yeah, the shell itself is made of silica, which is inorganic, and then you also have in the sample very often some carbonates, some maybe from some fragments of mussels, of snails, yeah, some sand material. So these are calcium carbonate, and that's also inorganic. And what I'm going to do now, or what I would like to show you, is how can you remove the organic substance, and that is you have to use a substance. I know some of you are going to be scared now. Hydrogen peroxide. Now it's H2O2. Will remove the organic material, and this takes quite a bit of time, so that's why I prepared something, right? And the carbonates, they can be removed with acid. Okay, if you add acid, then the carbonates are converted to carbon dioxide. Yeah, and basically when you do that, then the only thing, ideally the only thing that's left over are the diatom shells made of silica, because they are not removed by the acid and they're not removed by the hydrogen peroxide. Okay, so this is kind of the idea. That sounds fairly simple. Just add a little bit of hydrogen peroxide, and then later on just add a little bit of acid. After you remove the hydrogen peroxide, it should work. It does work. However, the digestion process of the removal here of the organic material takes quite a long time. It depends on the sample, but can take a couple of hours. So that's why I prepared something. Okay, so that is something that I would like to demonstrate with you, and I'm going to do two things. I'm going to actually show it to you here, but just to be safe, I actually prepared a video as well, which I'm going to comment. So that's basically the sample, okay? And you see the algae are kind of dense on the bottom here. And what I've done is just to, if this is not the case, you can also centrifuge it. I think this centrifuge is rather optional. So I've used it a couple of times now, but it's if you're patient and you allow the things to settle, then it will also work. And now you can actually see that the algae are much more condensed down here. Was not really necessary, I think. Okay, but what I've done then is I removed all of this liquid on top, to make sure that we have a concentrated sample of algae, and of course the diatoms are also down there in this what we call the pellet. Yeah, and then I added the hydrogen peroxide. Okay, I will show you which one I used, and then immediately I could see that it started to bubble, and these bubbles here are actually oxygen. The hydrogen peroxide now will essentially oxidize the organic material. It will actually digest it, so to say, and in this process it will form also oxygen. You got to be really careful with the hydrogen peroxide, okay, because it is quite aggressive. It depends on the concentration. I'll explain which ones I've used. Yeah, so now the video is basically looping again. Okay, so the thing is, what I'm going to do now is I'll just show you the thing. Can you add peroxide and acid at the same time? You got to be careful, because if you use acetic acid like vinegar or so, then apparently the hydrogen peroxide reacts with that and forms a pretty toxic substance. So it's actually the protocols that I've seen is that you should do it one after the other. Okay, so look, this is what I used here. I happen to have it. I got it somewhere, and this is actually, it's a little bit out of focus. This is simply a hand disinfectant. And I checked the ingredients here, and you can actually see that it contains hydrogen peroxide. And down here it says it's a 3% solution. Okay, so hydrogen peroxide is a 3% solution is very common for disinfection, and that's of course safe to put on your hands because it's only 3%. However, there's the disadvantage with that is it takes a long time. Okay, so it's more safe to use. Obviously, because it's used for disinfecting, it's more safe to use than look what I got here. Look what I got here. Yeah, it's a 30%. So this is 10 times as concentrated. Yeah? You cannot just buy this freely. You got to go to the drugstore and you have to possibly order it. Maybe they would like to know what you use it for. Yeah? But this is pretty aggressive stuff. Okay, so you don't want to get this stuff on. I remember once I got it on my fingers. I was still working at university in the lab, and I was not careful. I got a drop on the fingers here. It didn't hurt, but essentially it turned all white. And after a couple of days, the skin went off. It's like actually having a burn. Yeah, so you don't want to get this stuff into your eyes, right? So this is pretty aggressive stuff, and if you look carefully, I don't know if you're able to see this, but there's some actually oxygen bubbles forming as well. So this basically means that the hydrogen peroxide is unstable, and it will actually degrade and break down after some time. Yeah. So short answer. The 30% actually is 30% hydrogen peroxide and 70% water. Okay, I did a little bit of research that higher concentrations are actually also available, but they are only used for industrial processes. Yeah, and I think even it's also used as an oxidizer for some rocket fuels, and then I think you're using over 90% or something like that. Yeah, so this is not readily available, and the 30% is actually also the amount that the concentration that's written on the original on the container. Yeah, so the hydrogen peroxide always goes with percent. However, I think the percent, of course, will go down as the hydrogen peroxide ages. Okay, so that's a little bit the thing that I would like to show you. And what I'm going to do now is I've started. I started to use a 3% solution, and then you have to heat it up. How do you heat it up? You put it into a water bath, a hot water bath. So what I've done is I've used those test tubes here, not test tubes, these are actually centrifugation tubes made of plastic. And even though the plastic is actually also organic, interestingly, the hydrogen peroxide does not attack it. I did also some research why this is the case, because those long chain hydrocarbons in the plastic have too few so-called functional groups with which the hydrogen peroxide can react with. It's a little bit of chemistry. So the first thing that you want to do is, if you have a sample like this, is you want to remove. Look, I've actually got a pipette here, and the tip over here, I just realized, is a little bit too thin. So I'm going to cut off just to make it a little bit thicker. And I'm going to transfer some of the algae as far as this is possible. Now you're able to see it, yeah. As far as this is possible, I would like to transfer to this one over here. Okay. And there's one thing that I realized, not surprisingly, you have to be a little bit now, you have to find the correct amount. I will tell you why. If you use too much of the substance, then of course the hydrogen peroxide is going to be used up much faster, right? And it's not going to be quite as effective, and then you have to exchange the hydrogen peroxide more often. If you use too little of this, then the sample that remains is going to be too small, because most of this organic material here will be actually removed. So the actual final sample of diatoms that you're going to have is actually quite small. So I started off, just to show you a little bit, I started off with a sample like this, and look at the end product that I got. The end product at the end here is only that much. You can barely see it. Yeah? So these are more or less concentrated diatoms that I have here. Yeah? And I started off with a sample like this. Yeah? And that's what's left over after the digestion process. So I just want to tell you, we have to find somehow an ideal middle ground. If you use too much sample, you need much more hydrogen peroxide. It's going to take much longer. And if you use too little, then you're going to have too few diatoms. Yeah. So this is a little bit the issue over here. Yeah. So I'm reading again some of the comments. Okay, that's it's by mass or by volume. Let me check. So the question is, let me check. There is actually an original bottle that I have here. It says here hydrogen peroxide, concentrated hydrogen peroxide, 30%. They call 30% concentrated, and they include something to stabilize this. it says 1 kilogram, 30%. Yeah, safety instructions here. Use water to rinse. Don't get into the eyes, and so on and so on. It doesn't say whether it's per mass or per volume, and I don't think it really makes a huge difference. The reason being because the concentration is going to change anyway as it's used up. Okay, so that's the thing. And now you're what I'm doing now. You're not looking, okay? Because but the better way I have to I need some tissue paper over here to wipe in case I spill something. Okay, and yeah, I'm going to now use the concentrated one, and I'm going to add approximately the same amount here. Yeah. I don't know if you're able to see this. Okay. Always make sure that there are no drops. Yeah. So, and when you look at this here, I don't know if you're able to see this. Okay, you can already see that, yeah, the whole thing starts to float to the top, and the reason is because the oxygen bubbles, of course, pull everything to the top because the gas floats, and essentially you can see now a whole bunch of oxygen bubbles forming. Usually, what I do is I put a cap on here to prevent spilling. Okay, and I usually tap it a little bit to make sure that yeah, it dissolves properly. Yeah, and you see the reaction is starting. And I would like to explain to you now a little bit why is it foaming. Well, the hydrogen peroxide is now basically breaking down. It's forming so-called radicals. Radicals are highly reactive substances, and it kind of reacts randomly almost with the organic material. However, because there are now microorganisms in here as well, many of the microorganisms have an enzyme called catalase, which basically breaks down hydrogen peroxide and which forms then also the oxygen. So what we see is that when after the reaction the bubbling has stopped, and when I exchange the hydrogen peroxide again and add new one, it's not going to bubble quite as much, okay? Because the catalase is already has been destroyed. Yeah? So usually we find always at the beginning quite a bit of a reaction happening. Okay, and what you have to do now is you have to put the whole thing now into either you wait like this at room temperature, but then this is going to take quite a long time, or you put it into a hot water bath, for maybe an hour or 2 hours. Okay, so that's a little bit the thing, and every now and then because it starts to float, I try to knock a little bit on it, and then I hope that the whole thing is properly submerged. So I want to show you now. Let's say that this has been now in the for a few hours, and then I just want to show you how it starts to look like after a few hours, because I've prepared those actually today in the morning. Same sample, by the way, and look, the color has changed significantly, right? It's become much brighter. This one is a different sample of algae. It's become much more homogeneous because the things have been kind of digested. And also the amount decreased because some of the things actually have been oxidized, have gone into solution. And I did not centrifuge those. I simply let it stand, and you can actually see that it starts to collect on the bottom, and the bubbling has stopped. So what I do then now is I take a pipette, I remove the hydrogen peroxide, I replace it with fresh one, I mix again, and I let it digest again for a few hours. Okay, and you're going to see that the sample actually becomes less and less because more and more of the material is digested, and it also becomes whiter until ideally at the very end you have something that looks almost like some white sand. Yeah, like I have over here. Yeah, actually, I don't know if you're able to see. It looks almost a little bit like white sand, and these are then basically the diatoms. Ideally, I say ideally because it's in this case still not entirely pure, right? So you see that the sample is actually decreasing quite a bit. Okay, and I don't know where's the other one that I have. Essentially. No, I remember. This one over here I did today in the morning. And this one over here, that's the one I wanted to talk about. So this one over here I did it today in the morning. And this one here is a different sample with a different algae. And I actually made this one over here last week, so 1 week ago. And it still didn't properly digest. So that's actually one of the difficulties that I found is that different samples seem to be more or less easily digestible. Because this one over here, still is almost the way that it was at the beginning, but this one over here was actually was quite effective. Yeah? So evidently not all samples can be easily digested. Yeah? So I'm going to go a little bit here again. There's a so is 30 volumes 9% H2O to used for here, or truly 30% peroxide? Is 30 volumes? Well, the thing is the following: 30%. I cannot imagine that 30% peroxide is used for here, because 30% is quite aggressive. Yeah. So actually, I once did an experiment. I actually took some of my hair, put it into hydrogen peroxide. Of course, it turned white. My black hair turned white, but not only, but it actually started to dissolve. Right, obviously, because the hydrogen peroxide also broke down the hair. So there's some off-topic questions. So you centrifuge the specimen again after using the hydrogen peroxide? Yes, you can do that, but sometimes if you're patient enough, and I have centrifuged it, but if you're patient enough, then you can actually wait for the thing to settle on its own. Yeah, so I found this is actually also works. Yeah? Centrifuging is faster. Yeah, and in that sense more convenient, but because the diatom shells are actually much heavier or much denser rather, I found them actually to settle quite fairly rapidly. Okay. Can we use hot bleach instead? I've done a little bit of research, and I've seen that people have also been using also other oxidizing agents. However, the one with the hydrogen peroxide seems to be the standard procedure. I've even been thinking, because I don't know if you remember a couple of weeks ago, I put some dishwashing powder to ketchup because I wanted to digest this, right? Maybe this would also work. But hydrogen peroxide is actually the standard protocol to be used. Yeah? I'm just reading again. Yeah. So, comment here: hydrogen peroxide for hair is usually 6%. Yeah. Okay. And 30% is pretty aggressive. So what we're going to do now is the following. What time is it? Okay. Yeah. I would like to simply show you now. I mean, I've already done this with the other one here, but we're just going to make now some slides. No, you know what? Before I do that, I just want to show you something else. Just as a little interesting reference. These here are. Let's close this a little bit. Unfortunately, also started to dry up. That was a little too slow. These are actually. Here we go. That's actually diatomaceous earth. I can show you. I bought this some time ago. Look, it's because of the blue-green color, it kind of got transparent. This is I bought this. It's diatomaceous earth, and you can see also in here. Actually, it used to look a little bit nicer. Basically, the diatoms, not so nice. You know what I'm going to do is I tell you why it's not so nice, because it started to dry up, unfortunately. Yeah, I'll show you later. No, actually, I could show you now. If I add a little bit of water, if I can find my pipette, then let's have a look at how diatomaceous earth looks like. Let's try this. You are the desk view. You see, I prepared this before, and then so here we go. So I'm refilling it with water. Yep, that's better. Okay. So what you see, these are basically what is supposed to be, fossilized marine diatom shells and fragments. They look quite a bit different from the ones that I've extracted. I just want to show this to you as a reference. Yeah. Yeah. So that's a little bit, yeah, just for reference, okay? So, but that's not what I extracted. This was just simply in this container. And let's have a look now. And I would like to show you a little bit. Where are they? Those two were actually part of the same tube, right? And I separated this. I just want to explain to you what I've done here, because yeah, I'm just going to show you. Because they are a little bit different, even though they're from the same container. One of them settled first on the bottom, and the other one was basically still in the liquid above. And I'm going to show you now how this actually looks like. And I'm going to, for this purpose, take a sample from here. It's a very small amount is all that we need. I change the tips because I do not want to cross-contaminate. Okay, and here now I'm taking from the other one. Also a small amount. At least I hope that I'm able to show you that what I wanted to demonstrate. Cover glass goes on each one of them. Here's the other one. Yep, actually I think it might work. And let's have a look at the left one over here first. So that was basically after a couple of hours of or days even of extraction. Scope view. I need to do it a little bit smaller here. Yep. And this is basically, let me turn down the light a little bit. That is basically the, yeah, from the sample where it settled first. Yeah. And what you can see is a lot of sand grains, which is interesting because what I did not take the sediment from the bottom of the lake, but actually some floating algae, right? But apparently there was still so much sand in there, which yeah, probably also silica, right? Yeah, that it actually, yeah, the diatoms you are still able to see. There, of course, the diatoms in between. You can, if you look very carefully, then you can actually see them. Where is this? In here, right? Those shells, but there are these huge grains here. I treated the whole thing also with with acid, of course, and in order to remove all of the carbonates that are present, but they still remained. Those things here, right? So it kind of shows that maybe a way of filtering could be possible. Maybe, I don't know. Maybe by passing the whole thing through a plankton net. Now I'm going to show you the other one over here. Okay, and that is basically was from the liquid that was still on top, right? I separated this and waited for that to settle. So this was still stuff that was floating around. And we can see here that there are far fewer sand grains, actually none of them. There are many more diatoms, however, and this is a little bit where I'm only, let's say, 80 or 90% satisfied. There is still a lot of other stuff here floating around, which was could not be fully digested away. Yeah, even after a couple of hours of hydrogen peroxide treatment, which I think is a little bit of a pity because I don't know how to actually get it removed. Yeah? So this is the thing here, right? But you can, in any case, I think see quite nicely that the concentration of diatoms is quite high. So of course, way more than in the natural sample. So there is some lively discussion going on here about hydrogen peroxide. Let me quickly have a look. Yeah. So yeah, hydrogen peroxide, yeah, is. I don't know, it's just I'm just reading here. Yeah, it says, yeah, exactly. My bottle actually specified 1 kilogram and not 1 liter. So they were going by mass. Huh? Yeah. And usually, yeah. Did you say plankton net? Yes. Yes, I just said plankton net. There's something called a plankton net, which is a very finely meshed net, which you basically pull through water, a lake, for example, right? And then basically plankton, like algae and so on, are actually caught in the net. Yeah, it's a very, very finely meshed, standardized net. And I was actually thinking maybe if I could get one. I mean, I don't have a plankton net because I'm not sampling this way. For this reason, but I think if I'm going to get one, then I would actually probably have to cut it apart and make a filter out of that to be able to pass that through. Yeah. Okay. So in contrast, 30%. So there's a question over here: to what temperature should it be heated to quickly remove the organic material? And even that I tried out. I read online in the protocols, basically what they're saying is cooking hot. Okay, and I've done that. It's basically chemical reactions happen faster. If you don't heat it up, you just have to be more patient. Yeah, so that's all I want to say. Yeah, and what I've done is I simply placed it into a glass with water, into a water bath, and then basically kept it at a high temperature. So this is something that I really recommend. I remember a couple of years ago I've also done that, and I tried to extract the diatom shells from the sediment, not from the algae that were floating around or from scratching material off a rock. I tried to get the algae from putting some sand from the pond with hydrogen peroxide. This was actually not very successful because there were so many sand grains in there that it was very difficult. Yeah, and then you actually have to filter it. Yeah. Hydrogen peroxide. Yeah. So yeah, I know that for example, when we buy the hydrogen peroxide, I say we because I'm a teacher in school, and for some experiments, or demonstration experiments, we also need them. The drugstore that sells us the things already knows us. Otherwise, they're really reluctant sometimes of selling you these things because they're kind of worried that you do something stupid with them. Obviously, because if you spill them, then actually, you don't want to get this stuff into your eyes. Yeah. So this is, but 3% hydrogen peroxide, I've also tried. It also works, but just much slower. Okay, so this is a little bit the thing here, where I'm still kind of wondering a little bit is how can I get all of these other things removed? So for example, just show, here, that's kind of interesting. Do you see those regular patterns over here? I don't know. Almost looks like these are cells here. Yeah. So if this is some kind of some leftover of a plant, then it was not properly removed by the hydrogen peroxide. Yeah. So this is something where I don't know how to do that. Yeah. I've also read somewhere that some people have removed all organic matter by actually really physically burning the diatoms. Because if you burn them, then you're burning away the organic material, and what's left over are the shells of the diatoms. So apparently this is also a possible method. Yeah. So what happens to the microplastics is a question over here, and I think that nothing will happen to the microplastics. I think they might not be removed because the plastic tube itself that I have here is also made of plastic, and the hydrogen peroxide does not attack it. I did some online research, and apparently, and indeed, it's stable. And also the container over here, is in plastic, with the hydrogen peroxide. And apparently the reason is because the plastic has these long hydrocarbon chains, which are extremely stable. And there are few functional groups that the hydrogen peroxide can actually attack chemically. So I assume that the possible microplastics in the sample will also not be attacked by the hydrogen peroxide. My assumption. Yeah, but then again, there are so many different types of microplastics out there, so I guess it depends which one it is. Yeah. Is it possible to heat mount the diatom shells after concentration? They are so small that I would prefer to use my oil objectives. Ah, I get what you mean. No, I wouldn't. So that okay. I need to explain this a little bit. The question is it possible to heat fix the diatom shells? And my assumption is probably not. What happens when you heat fix it? I just show it to you over here. And why I think it's not a good idea. What you do over here, that's basically, these are some diatoms and I dried them. So what you normally do when heat fixing, and you do that with bacteria, is that you pass the slide through a gas Bunsen burner, and this causes proteins to basically cross-link with the glass, so they stick to the surface. Because I've treated the whole thing with hydrogen peroxide, I've hopefully removed all of those proteins. So there is simply not enough organic material left or proteins left to actually cause the diatoms to stick to the slide. That is my hypothesis. So I think it might not, heat fixing might not work. However, what you can do, if you want to use oil immersion, is why not simply apply a small drop of water here and simply put a cover glass on top and then still use oil immersion. So I think that should not be a problem. Yeah? Or you try out whether to what extent actually heat fixing does work, or whether it's stable enough that the diatoms don't come off after you've applied oil on it. So that's something you might have to experimentally try out. But I would probably guess that heat fixing might not be, I'm guessing, might not be the method of choice here. Okay. Okay. So let me have a look here. Yeah. Here's some discussion going on about the plastic tubes. I think they're polypropylene, right? So I think they might be quite stable. So, okay, there's some microscope eyepiece questions. Diatoms make their shells out of silica. Can we call it glass even if it is made of really low temperature? I mean, I think it's silicium dioxide. Some books actually say that, yeah, diatoms have shells made of glass. Yeah, so I guess it wouldn't be that wrong, because chemically it's actually the same thing. Yeah. I'm just reading here. Okay, yeah. So let me quickly check. Let me quickly update this here. Okay, there. I was just checking how many people are watching. Okay. Yeah, so having said that, let's have a quick look back at I'm also checking the time. And I would like to try. Okay, so that is basically the wet mount. Okay, so that's basically in water, and you can actually see that, yeah, there's still, you know what, if you press against it, then you can actually see that the whole thing is still in a liquid. I'm going to press against the cover glass. Yeah. You see, the whole thing still seems to be sticking together a little bit. Yeah. Look, there is a... Could this be? I don't know. This long blue thing here, that could be actually a microplastic, what we have here. Yeah. I don't know if this was already part of the sample or still on the microscope slide. But these things here that you see here, especially if they have some kind of a blue color, so sometimes these are actually microplastics. Lots of these fibers from clothing and so on can be found in natural samples. Actually, chances are pretty good that you're going to find them. The chances of finding them are higher than not finding one in a sample. Yeah. So what I'm going to do now is the following. I would like to now mount a couple of slides here in two different mounting media. So basically, this is something I would like to try to do. So, okay, and let's try this. Where is the desk? Because the refractive index is really important. There are actually certain mounting media that are actually recommended for diatoms. Because, as we just mentioned, they're kind of made of glass, those shells. If the mounting medium has a refractive index, it's so similar to the diatoms, then you're not able to see them. Okay, so that's a little bit the problem. Yeah. So what I would like to try, and again, I've not tried this before. Sometimes I do like to experiment around a little bit. Is I'm going to use Uperol, which is actually look at this. This expiry date is 2013. It still works. It still works. Okay. So let's try this here first. Let's give it a small drop here. Yeah. So, by the way, as I'm making this, a couple of weeks ago, last week and 2 weeks ago, some of you have thankfully financially supported this by donating a little bit of money. I just want to inform you that I've ordered some more antique microscope slides, and they're currently being shipped. They're now on the way, and when they arrive, I would like to show them to you again. As a matter of fact, I've ordered 7 slides with the spines of sea urchins. I found some nice ones. There's several more of them, and that's another thing that I would like to figure out. How in the world did the people prepare the spines of sea urchins, because they're so extremely hard? Yeah. So let's put it on here. Let capillary action do its job. And yeah, normally you got to be really careful. Normally you have to wait until the whole thing dries properly before you put it under the microscope, because during the drying process the refractive index also changes, and you do not want to get any mounting medium on your microscope objective. But there's nothing, no mounting medium is spilling out. Just see that the cover glass is a little bit dirty. But what I would like to try now is yeah, that's basically in water. Okay, and let's see how this now looks when we use a different mounting medium. Okay, so here we go. I have to refocus again a little bit. And this is how it looks with Uperol as a mounting medium. Okay. You see that apparently the refractive index is obviously different, so the diatoms look way more transparent. Huh? So the refractive index of the mounting medium is quite important. Let's go up with the magnification. That's now 60 times. Let's refocus again a little bit. Yeah, I've got DIC. You know what? Let's have a look how does this look without DIC. That's basically regular bright field. I actually might want to try out. Oh, thank you again. Thank you very much. See, I got another donation here from Paul, and yeah, thank you. Yeah, so that's actually looking at this. This actually does also look kind of pretty. The problem that I have is I've unfortunately dark field only for my 4x objective. Not tried that. So you know what? Why not try phase contrast? Let me see how phase contrast looks like. I don't know. Ooh, that's nice. Actually, that's really nice. That's phase contrast. Look at this. This is, oh yeah, I'm happy. That's to the condenser close too much. So that is phase contrast as 40 times. This is actually quite nice. Yeah. No, seriously, I've not tried this yet. I'm going to now, that's with the Uperol mounting medium. Let's have a look how this actually looks like with the water from before. From the water from before. Where is it after? Oh, where is it? Here it is. That's interesting. Look, I mean, it's a little too dense, maybe. Here, maybe that can also play a role. That maybe, but look, the diatoms seem to be darker. Here, some of them. Huh. Let's go back to the other one. That's again the one in. I have to find the focus again. In Uperol. Yeah, actually, this looks kind of nice. Yeah. So, okay, well, good. Let me see. 4 times. This would now be dark field, but I think the diatoms are a little too small. Nah, see, you don't see. you don't see. Nope, that's too small. Yeah. So yeah, let's go here. Exactly. You can. That's the 40 times again with. Where is it? 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 Here we go again. Okay, we're this again in phase contrast. Okay. Well, quite nice. Quite nice. Let me quickly have a look again. Just a second. That's the reason why it's okay. So, quickly checking again some of the questions. Yes, don't seem to be anyone. Okay, so you know what? I would like to now try something, another one. And where is this the desk view again? So I've got another one over here, which basically I already prepared dry. And you know what I'm gonna do now? This one over here, right? This is PVA glue. Yeah, if you basically have been watching this stream, then you already know that I've been using this quite frequently. This is a basically water-based glue, which I diluted with more water, and it's Elmer's PVA glue, which I bought over very cheaply over Amazon, and I diluted it with water, and yeah, it's good enough for general purpose work, and I'm going to try it here as well. I assume that it will have again a different refractive index and again different properties we're going to see. Okay, so, and again a cover glass goes on top. So I'm just having also a little, how do you say, look. Okay, still a little bit too concentrated for my liking. Yep. Yeah. So that's a little bit of thing. When it's too concentrated, you want to add a little bit more water, and then you might want to, how do you say, spread it a little bit more. So let's try this one here a little bit. Yeah, we have to be careful. Again, something spills out here. We have to be careful that we're not, rotating the objective into this scope view. So there's another question over here which I'll respond to in a second. I have to remove the two slides here. So this one was the one with U-Pearl. I'll have to remember that. This one is the one with water. I'll remember that. And this one over here is now the one with the PVA glue. And I'm going to start again using the lower power. Okay. Let's see. It's a little too bright. So. Lots of bubbles. And I don't really. That's interesting. Okay, maybe go up a little with the magnification here. Now that's, oh yeah. This sample seems to be, I mean, it's the same sample that I used, but it looks so, it's so different somehow. What it could be, and I do have a hypothesis, where there are far fewer diatoms here than in the other sample. I mean, there are a couple here, and I think the reason, I mean, I do have a suspicion of why this is the case. I mean, here, a few more. It can be that when I took out the, it's actually from the same tube, but it could be that when you take out the sample from the tube, because I allowed it to settle, that different basically diatoms and sand grains and so on with a different density, they of course they settle at different levels. It could be maybe that for one of the slides, maybe I took the sample a little bit more from the top, and in another one maybe a little bit more from the bottom, because as it settles down, you have a separation happening. So this kind of could explain why there are maybe fewer of them in here. It's my guess. Let's go up here a little bit. I mean, yeah. I mean, Interesting, interesting, and and you see over here, these are all diatom shells, but the glue isn't the right one. I think the glue is not the right medium. So what I do now is let's try again, phase contrast, just for the fun of it, to see how if there's a difference. Yeah, no. Yeah. Look at this here. I mean, this here is one of these shells here. Yeah. But it simply doesn't look as good. Yeah. So let's try compare this again with the other mounting medium, with the U-Pearl one. You have to refocus, of course. I don't know. This is a huge difference. Okay, the specimen generally is better, but the diatoms look better. Yeah. So I'll quickly read again some of the questions. So did you use an acid? A question to me, did you use an acid, or is this another option to sieving? Well, what I've done is, for those of you just joined in, I used hydrogen peroxide to remove all of the organic material from the sample. And then I did an acid treatment indeed to remove the calcium carbonate. Then basically I had at the very end, the diatom shells are the only things that were left over, or almost the only things. And so this is kind of was a little bit the idea. Yeah. So, and you use double cover slide on the same slide. Do you use to do this, or is it related to my suggestion a few weeks ago? I always do that. Okay, so basically the question was the following over here, that I've got essentially two cover glasses on the same slide, and generally, occasionally I do that. Yeah, especially if I want to compare samples, I usually put them on the same slide, because then I simply can basically move over the slide and can compare very quickly. Yeah, I basically have to do all variations. Sometimes only put one of them on here, sometimes multiple ones. Yeah, so usually what I do is I want to pick up something with a pipette, for example. Yeah, like this. Like a small specimen, and I usually transfer it over on the same slide next to it. So, are both of these mounting media permanent? Will the slides last? Yes, both of them are permanent. I will tell you, this one here is the U-Pearl mounting medium is commonly used. It's difficult to obtain though. But it is a very common mounting medium, especially used by people who are preparing insects. Why? Because it it makes the insects more transparent. But because it's kind of expensive, a little more difficult to get. For this reason, I was looking around for hobby purposes and for use in schools and so on. I simply bought online over Amazon Elmer's PVA glue, the clear glue. Added a little bit of water. And for many samples and simply for experimentation, this is also fine. Yeah. Different mounting media have different advantages and disadvantages. The glue has the big advantage that it's water-based. Ooh, I'm sidetracking a little bit, but it's fine. Yeah. But both of them are permanent. Yeah. Okay, maybe use acetone to get greater refractive index difference. That could also be. Yeah. Which acids did you use? What I did is I bought myself. So first of all, you got to be careful when mixing hydrogen peroxide. Let's put this picture back in place. When mixing hydrogen peroxide with acid, I read you have to be careful, because if you use vinegar, the hydrogen peroxide will react with the vinegar, and then you have a product which is a little bit harmful. Yeah. So what you should do is you should remove the hydrogen peroxide first, wash the specimen, and then you can add the acid. And what I did is it's not regular vinegar, which has about 3 to 4%, but they also sell in the supermarket very concentrated vinegar of about 25%. It's not, so, and I've used that, but regular vinegar should also work. In reality, actually it shouldn't make a difference which acid you use, because calcium carbonate will dissolve in acid. So I've even been thinking about maybe using a descaler for coffee machines, because they're also very effective in removing calcium carbonate. So maybe that would also be a possibility. Not tried it, but yeah, possible. Yeah. So that's it's actually quite nice. This one over here, I have to tell you. Another another thing that I can say is that what some people are actually doing is if you look at the diatoms very closely, then you can see that there are these, how do you say, regular patterns, those lines, yeah? And whether you're able to recede those lines or not, or resolve those lines, can actually be used as a measure on the quality of your microscope optics, because some of those little lines are kind of very difficult to see and very close together. Yeah, so some people are actually using this as a kind of a quality reference or benchmark for the microscope optics. Yeah, so I'm quickly going through the whole thing. Yep. Yes, last comment here, that is also correct. The official protocol says that you should use hydrochloric acid. Okay, it's an inorganic acid, so I can imagine that it maybe reacts also more neutrally with the hydrogen peroxide. Yeah? So hydrochloric acid is actually the official one. I didn't have hydrochloric acid at home, so I used this concentrated vinegar. Yeah. So that's basically it. I'm waiting for my assistant to bring me the questions. I hope he didn't forget about those. Otherwise, I'm just going to go through the chat myself and try to respond to those questions that are not topic related, because there were some questions about microscope optics as well. Okay, so let me see. I tried the famous onion experiment. Yep, here's a question. When you use your centrifuge, I noticed in your videos that you use the plastic tubes, these ones here, right? Yes. And not glass tubes for the specimens. Here's something coming. Thank you very much. Here are the questions. Cool. Wow. Heavens. A lot of questions again. Thank you very much. I don't know. You know what? I'll switch over to the desk view again. Okay, here we go. Let me move my microscope over a little bit. When you use your centrifuge, I noticed in your videos that you use plastic tubes and not glass tubes for the specimens. Is there a reason why you're not using glass tubes? Yes, there is actually I do have some glass tubes as well. There is not a super important reason, but there is a reason, and one of the reasons is that the test tubes are basically have a round bottom. Actually, I think it doesn't really matter which ones you use, but I like the pointed ones because essentially, the specimen is concentrated to a point down here. And also, it's just convenience, really, because sometimes I simply collect the sample directly in those plastic tubes. I just put a cap on it, I take them home, and if I want to centrifuge them, I just centrifuge them. So I kind of used those plastic tubes also as a, how do you say, storage container for centrifuging and simply, for convenience reasons. And if it doesn't become too dirty, then I, of course, rinse them out as well. But there is no, I would say, no specific reason. I like to use plastic tubes generally because, otherwise, if the glass test tubes break, then, of course, danger of injury. So, but I don't think that it's, there's no otherwise specific reasons. Off topic, I made a few slides with PVA water mix. Oh, okay. So suppose you use this one over here. But I noticed that the cross sections, the ivy leaf stems, shriveled up. Any ideas on how to avoid this? More water in the mix? Possibly not. That's interesting. What I probably would suggest is the following. Again, I'm not always familiar with all specimens. If you use a lot of water, and if the water evaporates, then it's going to shrink more. And it could be that maybe it was indeed too liquid. Okay, so try less water, because then the glue might stabilize the specimen more. But if you use a lot of water, and I think 1:1 is quite liquid, right? Then the water will evaporate, the glue will remain, the glue will have to shrink. And this might maybe also cause the leaf to be pulled together and shrink together, right? So yeah, so I would actually probably try it with less water. Okay, number one. Number two, think about or consider maybe putting the leaf as such into some of the mounting medium, maybe for a day or two, to allow the glue to soak into the leaf properly, so that when it dries, that essentially the glue is able to support the cell structure. Okay, so that's another. So maybe give it more time for the mounting medium to infiltrate, so to say, to infiltrate the specimen. Yeah? So I have only very rarely found diatoms in my. Oh, I already mentioned that. Okay, collect some solid material and also from surfaces, because many diatoms will actually like to grow on surfaces. Okay, not so much in the free water, because they're gonna sink. How do diatoms divide? Yeah, they have this basically nested division, so that basically you have a shell and then it forms a smaller shell within that, and so on. And then somehow, I don't fully understand myself now, it will regenerate this, and then you'll have again an original size. Yeah. In the previous question and answer session, you mentioned that methylene blue stains acidic structures in a cell. What does eosin? Oh, I don't know. Is it used for staining plant cells? I don't know which substance, which chemicals eosin stains. I'd have to basically pass it to the community. Okay, so please, if anyone is familiar with that, okay. Good evening. There's a vintage microtome for sale on Facebook. Are there things to look out for when buying? I mean, honestly, to look out for, yeah. There is one thing to. I mean, okay, I once bought something indeed over announcement over Facebook, and the thing that I basically, I tell you what the problem was that I had. I think it was a microscope objective. Basically, I got in contact with the seller and I transferred over PayPal. I transferred the amount, and then the whole thing got stuck in the on toll. How do you call this? Import, and they wanted to see a purchase receipt and an order form from the company that sold it. And of course, I didn't have anything like that, because it was simply a verbal agreement or text agreement, right? So I didn't have an official printout of an official bill or anything like this from the company. So they gave me some problems. So this is a little bit an issue that I have when buying things that are probably not so cheap from other countries, because you don't have any paperwork. And then it got stuck, basically at import. That's the only thing that I can actually tell you over here. I don't know, probably you wanted to know a little bit more about the microtome itself. Generally, if the microtome is in good condition, then why not? I mean, it's a mechanical device. Microscopes can also be quite vintage. However, I probably would not, how do you say, use it if it has some kind of historic value. Generally, there are sometimes historic equipment should be put into a museum and not be simply used in the lab. If it's a collector's item, I also have to say that. Okay, so that's simply a thing I just also want to mention. Do you use household strength hydrogen peroxide 3%? Yeah, I mean that's the one that I used over here, right? This is a disinfectant which worked a little bit slowly, and that's why I got myself a stronger concentration. Can you add peroxide and acid at the same time? I already mentioned this, that you probably, some people have been doing that, but you should not use vinegar. Okay, acetic acid, because acetic acid reacts with the hydrogen peroxide in a way that you have harmful byproducts. But apparently acid, maybe hydrochloric acid, I read, will actually activate the hydrogen peroxide more, that it will actually work faster. So we talked about this already. I worry about the sewage human flow into the Earth's seas with the human population as is. Could I ask, is the sewage human feces bad for the ecology of the seas? A short answer. Okay, I'm going to give you a, how do you call this? I'm going to deliberately give you a scientific answer now. Okay? Short answer would be, it depends on the amount. Okay? Because sewage produced by mammals in the water and so on, of course, will be broken down and decomposed. No question. So now, let's say that we're talking about extreme sewage, and now here's the scientific answer. And I'm going to try to answer this using evolutionary biology. The ecosystem, the animals and the plants in the ecosystem are adapted to a certain environment. Okay? And then all of a sudden, within a short time, you're massively changing the environment around, be it by sewage, be it by climate change, be it by pollution, be it by acid rain, it doesn't matter. If you change the environment around in a very short time, then the plants and animals and generally the organisms do not have enough time as a population or as a species to adapt to changing environment. Okay? And then there's a problem. Yeah. So what I'm saying here right now is if you increase the sewage slowly over thousands of years, okay, then the living things, the flora and fauna, and the plants and animals and whatever have time to adapt to the changing environment. Yeah, and they basically as a species will adapt, or the less adapted ones are going to die off. The better adapted ones as a species, they will survive, and basically this is what happens in nature. But if the change is so rapid that the organisms are not able to adapt, then basically they're all going to die off. Yeah. So short answer. Of course, we've got massive plastic in the oceans and so on. Yeah, and it's a question then, of course, is nature able to handle it, or is the quantity so much that it's not able to handle it? Yeah. So this was a little bit more the how do you say, the scientific approach. I don't know if this is actually what you were interested in, but yeah. So that's again H2O2. I think we discussed about that. Okay. Can I use methylene blue and eosine at the same time to create multi-stain specimens, or do both stains cancel each other out? I've never tried both of them at the same time, but generally what we do in differential staining, I'm going to avoid giving an answer. My instinct would be use it one after the other. Because you don't know whether they're going to directly interfere or not, and it doesn't have to be the stains necessarily, but also the solvents. Yeah? But then on the other hand, I would say that maybe the specimen itself will have a stronger impact on this. Yeah? So it's not only methylene blue that you have here, but usually it's in combination with, for example, Löffler solution that I have is in combination with potassium hydroxide, and there's also alcohol in there. It's rarely pure, and so I would say it depends a lot on the purity, or if the stain is already a ready-made mixture that also contains other substances. Yeah? Generally, what we've been doing at the university, we've been staining them one after the other, and not at the same time. Yeah, you simply have more control over it by doing it one after the other. But I would say that I'm quite sure that there are some protocols out there where it's possible at the same time. Yeah. So do you centrifuge the specimen again? I did. Or you basically, if you don't centrifuge it again, you just allow it to settle, and then you exchange the hydrogen peroxide. Yeah. Okay. It's used to this color here. Yeah, 3% is called 10 volume. Okay, so that's another. Yeah. So, I finally managed to get hold of small bottles of Uperol. What is the correct way to use it? Okay, yeah. Uperol has a couple of advantages over other mounting media. So first of all, the thing that I like about Uperol is that it's pretty long-term stable. And it has the following advantage, that you are able to transfer, you use it for two types of specimens, either completely dry, okay, That's one possibility. Or you have the specimen in concentrated alcohol, or for example, isopropyl alcohol. And then you remove all of the excess alcohol. It's still moist because of the alcohol, the specimen, the little fly maybe, or whatever you want to mount. And you can still take that specimen and mount it in Uprol, even though it still contains a little bit of alcohol. Uprol is able to handle that. Yeah. So if you have a moist specimen which contains water, then you have to make sure that you remove that water by putting in the alcohol, but the alcohol, a little bit of alcohol is fine. And as a matter of fact, I even diluted this one over here with a little bit of alcohol. It is also possible, and that's actually the better way, is to use the solvent, which is called Uprol essence. Uprol essence is a liquid, which is actually the solvent for Uprol, and it's actually better if you put the specimen into Uprol essence first before you mount it and put it into Uprol. But it's not necessary. I've never done that before, but that is the ideally recommended way. When you put the Uprol on the when you make a slide, you have to make sure that the slide is stores horizontally for about 6 weeks, 6 to 8 weeks. Uprol dries very slowly, and that is good. The reason is because it gives it enough time to actually soak into the specimen. Okay. Otherwise, Uprol is fairly easy to use. You want to keep it ventilated because it smells. It smells kind of pleasant. Like trees, freshly cut trees, otherwise easy to use, and just make sure that you give it enough time to properly dry. Yeah? So plankton net, yes. That's the net which where you can basically sweep it through water, and then you can collect plankton. Again, the percent H2O2. Yeah. It's easy to get 12% on Amazon. To what temperature should it be heated? Yeah, I've mentioned this already. It can be boiling hot. Yeah, and the hotter it is, the faster it will work. Yeah. So, is it possible to mount the diatom shells? I mentioned this, that I probably would not do that, not tried it, but I assume that the diatom shells are probably not going to stick to the glass. Okay, I have to try it out. What field of view angle of an expensive eyepiece can I get? Okay. You don't use degrees really. Love to get even wider, like telescope ones. Okay, I'll be honest with you folks. This is a good question, and I've been thinking now for already a week or two to actually, for my second YouTube channel, to talk about the field of view. Okay, and the field of view, and I don't have an eyepiece. Well, actually I do have an eyepiece here. Just gonna take it out carefully. The field of view also depends on the diameter. Okay, here. And this one has a massive field of view of 26.5. Yeah. So that's basically the value. And this value cannot be bigger than the diameter here. So this is a 30 millimeter eyepiece. And essentially the field of view depends also on the diameter of the eyepiece barrel that you have. Yeah. So, but with degrees, I have to tell you, I've never used degrees. Yeah. So it's the field number. Yeah. My 25 to 24 millimeters. Okay, that's 24 millimeters. If that is the field of, okay, that's interesting. Yeah. So normally the eyepieces have a diameter of either 23.5 millimeter. Oh, that's the 24 millimeter. Okay, and then you have to check the field number, and you can do that actually by measuring out the lens diameter. Okay, on the bottom side. You know what? I'll make a video of this. Okay, because this is something I wanted to talk about anyway. Diatoms make shells out of silica. Can we call it glass? Actually, some books actually do that. They call it made of glass. This purification, does it separate both halves of the shell, or do they stick together? That's actually a good one. I don't know exactly. Well, actually, I read the following. I read that the two shells are stuck together with some kind of an organic glue that the diatoms use, and hydrogen peroxide has a difficult time to actually break down that glue. Maybe it's because it's hard to reach. So my assumption would be that essentially if we allow it long enough to work on the shelf, then probably it's going to dissolve it, and the halves are going to separate. Yeah, I even read somewhere that some people have been using ultraviolet light to speed up this process. Yeah, also something I've not tried, but apparently. Can only repeat that what I read myself doing research. There is a glue that holds the diatom shells together like this, and the hydrogen peroxide has a difficult time to digest that glue for whatever reason. Yeah, so my suggestion would be it depends a little bit maybe on the temperature and on the time and on the concentration of the hydrogen peroxide that is used. Okay, for this one over here. Please do a follow-up to invite multiple people with a microscope and camera for an eventual live event. That's already been suggested, and I really like this idea. I have to see how we can do this technically. I know already some have a small YouTube channel and proper camera and skills. I mean, that would be really nice if there were somehow a possibility to kind of link everything together. I mean, what I could do here is if people go live at the same time, what I could do is I can open the different live streams of the different people, the windows, and kind of combine them into one window here and then start to live stream that. That could be a possibility. I've not tried this yet, but I know that the software can do screen grabs and screen captures. So if basically multiple windows are open, each one of them with a live stream where you show something, then I might be able to combine them together into one view so that the viewers can actually see multiple screens at the same time. Actually, a really nice idea. I still have to figure out a way how to do that. But please let me hear it if there's general interest in that. Then we can actually do that. Would be really nice to kind of for the community. Do you know why a way to permanently mount algae? I mean, not shelled like diatoms. Yes. You have to use algae are difficult. Algae being are very delicate, and if as soon as they dry, they shrink. So what you have to do is you have to for algae you commonly use glycerol jelly or glycerin gelatin as a mounting medium. And this one is difficult to use. You have to heat up the mounting medium. It melts when it's warm. If you make it too hot, you're gonna break it. In other words, it's not going to become solid anymore. So you have to warm it up to 45 degrees. You mount the algae and then you cool it down. And then it becomes semi-solid because there is still a little bit of water in the mounting medium because the algae, if they are not allowed to dry out completely. Yeah, so this is indeed, and then you have to use a ring. You have to make a ring around the slide to prevent complete drying out. It's more complicated. Yeah, so algae can be what I have done as well. I have mounted algae in this one here as well. It also works. So I probably would try that first, but because it's water-based, algae and so on should not be mounted in mounting media like Uperol or so, because the solvent will actually dissolve the chlorophyll, which is the green pigment of the algae. So what you actually see is that the algae will lose color when you use a mounting medium that contains an organic, some solvent like Uperol. Okay, and that's why water-based mounting media are ideal. However, the PVA glue might also, because it dries up completely, the algae might also shrink. Therefore, I've tried it. It kind of works, but glycerin gelatin works better, but it's difficult to use. Okay, because you have to melt the mounting medium, you have to put the algae in, you have to put a cover glass on top, it has to dry, and then you have to stabilize everything by making a ring around the cover glass. Yeah, because the glycerin gel does not completely solidify; it stays semi-solid. Okay, so that's a little bit the thing here. So it's a slightly more advanced. Okay, so what I'm gonna do now is I'm going to quickly I'm quickly going to look at if there are some remaining questions or comments. Okay. Yeah, my other 10 times objective is a 18 millimeters and it's about 50 degrees. What is the widest view we can get with an expensive ones? Let's put it this way, the ones that I have a field of view of 26 millimeters. Okay, 26 millimeters, and that's a more expensive one. But you have to make sure that what I've seen is that there is one that has, I think, for 24 millimeter diameter barrel, 23 and a half millimeter barrel. I think the widest ones that we can get is around 20 or so, 20 or 22. So let me quickly go through the questions here because it's already 1.5 hours. Let me quickly. I now have to find the place where we stopped. Okay. So I have to find the place where I stopped. I can recommend a stain. My preferred one is neutral red because it is pH sensitive and gives the information a nice color. Okay, thank you for the information. Neutral red stain. I would love to learn more about various sample preparations and purification of stain. Okay, thank you for the comment. I would recommend either Discord or Jitsi to do a multi-stream so there is no delay between communications. Ah, I see. Okay. Discord or Jitsi. Discord I know. Jitsi I don't know. I'll have a look into that. It of course also means that the people have to have an account for these platforms so that they're able to stream. Does anyone know any good forums about microscopy? You can actually check out my forum. Yeah, if you go to the webpage, microbehunter.com, there is a link to the forum. I do have to tell you that I'm still struggling a little bit. Sometimes the web page is still a little bit unstable. It gives you sometimes an error message. You just have to press the refresh button. I'm still in contact with my provider. I'm upgrading, but it's improved, but a little bit. Sometimes I'm still getting the error messages. But if you want to have a microscopy forum, but please, if you want to use the microscopy forum, you have to send me an email. Otherwise, I'm getting so many spam registrations. You have no idea how many I'm getting. It's really a lot. So you have to actually register an account in the form, and then you have to send me an email, and I'll try to manually unlock you. Because the spams are just unbelievable. Okay, and I want to keep the forum reasonably spam free. Yeah? My other 10x is okay. Yeah, we talked about this. Check. Oliver, advantage of Jitsi doesn't require an account as it is open source. Ah, that's interesting. They just join in their browser. I could help with hosting and setup if needed. Thank you very much. Okay, I'll have a look. Okay, I'll have a look into Jitsi, and I have to figure out how to do this. So then maybe I can also give some post somewhere some instructions so that other people who would like to do that can actually join in. And thank you very much to Michelle for another donation here. And as I mentioned, I'm using those donations to buy microscope slides, which I'm going to of course show you. Thank you very much. Yeah, is it the email through the forum? So basically, what you do is you send me an email to editor@microbehunter.com. Editor@microbehunter.com. It comes to me, and then I'll basically unlock you. Please tell me your username or your email that you used for registering. You also have to basically answer a couple of microscopy questions, and there's also a justification that you have to write. Why do you want to join the forum? And the reason why I've included that is because spam bots generally don't give or in the past have never given a proper justification. I just want to join, or I want to learn more, right? But if you provide a little bit more info, then I actually know that you're a real human being. That is actually the point why I have these questions there. But now with AI, even AI is not able to answer those questions. So we've had, again, more and more spam registrations. And for this reason, I got to do this manually because otherwise it's becoming a problem. Okay. Yes, thank you again for the donations, and I think I'm going to call it quits for today again. It's been again 1.5 hours. I hope that you enjoyed it. at least a couple of personal words from my side. That yeah, I like doing those live streams and videos and so on. Why? Because I myself also have to try out new things and new preparation techniques. So basically, these live streams are also a good motivation for me to try out a few things. And usually, what I do is to keep also the excitement up for myself. I do not always prepare everything in very much detail in advance. So I also like to be surprised every now and then. So for this reason, for example, the nice pictures that you see now in the background, I've not seen before myself. I've not observed this specimen using phase contrast after I've prepared it. So I like doing these things. I hope that you do as well. And I would like to also invite you, and that's another thing before I quit. This channel is currently stagnating. It's really getting stuck. Views are getting stuck, and it's not so much about the channel, but actually I would like to promote microscopy as a hobby. I would like to promote nature observation and so on. And currently it looks like a little bit that there seems to be less and less interest, or I don't know, or the algorithm, the YouTube doesn't promote the video. I don't know. So I would like to invite you, please subscribe to the channel, because this also is a sign to other people and maybe also to the algorithm that this is a hobby that people like. And then I hope that more and more people are going to start joining and picking up the hobby. The community. So that's a little bit the thing that I would like to request. And I will have a look here again. Okay, enough for today. Have a nice, still have a nice weekend. Hope to see you again. And I'll think of something else again for next week. Bye bye. Happy microbe hunting as always. See you around.